Hello, everyone. Welcome to BrainMap. This seminar series is co-sponsored by the P41-funded Center for Mesoscale Mapping housed in the Martinos Center. It is our great pleasure today to have Dr. Fei Du for our BrainMap talk. Dr. Fei Du is neuroimaging physicist, the director of Laboratory for High-Field Imaging and Translational Neuroscience at McLean Hospital, and an associate professor in Harvard Medical School. He earned his PhD in NMR quantum physics from the Wuhan Institute of Physics and Mathematics, Chinese Academy of Science, followed by the postdoc training at CMRR, University of Minnesota. Before joining McLean Hospital, his research focused on integration of MRI, MRS techniques, developments with translational neuroimaging aiming to outline biological abnormalities and trajectories and identify treatment, engaged target, and biomarkers for disease progression and treatment in neuropsychiatric disorders. I would like to remind our audience to address their question by raising your hand or in the Q&A box. Thank you very much, Dr. Fei, and the stage is yours.
All right, okay. All right, thank you so much, Chehu, for the introduction. And also a great, my pleasure to come here because I have a lot of collaborator colleagues in this image center. We just meet with Eva and Jensen a long time. We want to do some collaboration here. So really a great appreciate for this opportunity. We can share my experience, what I did, what we could do in the future. Okay, thanks so much. So today, I would like to talk about something about molecular mechanism underlying disruptive brain security in psychotic disorders. How can we use multimodal neuroimaging approach to figure out this problem? Again, my most work is psychotic disorder. That means schizophrenia and bipolar disorder. Yeah, that's I work in McLean Hospital. McLean Hospital is a specialized hospital for psychiatric disease. But really, I would like to mention about it. Okay. Yeah, that's good. So I really want to mention about it. All the method we could talk about could be used for neuropsychotic disorders. That means include neuro and psychiatry, right? Okay, if we talk about neuropsychotic disorders, immediately we could think some keywords. First, cognition function, right? Second, security, network function connectivity. Because always we work with mental health or psychiatry people, oh, your brain connectivity is not good, or some other with brain security system, right? Another easy thing about it is brain structure. Because brain function depend on your structure, fiber orientation, or one location to another location, right? This one. Another, I think we have to deal with molecular mechanism too, right? That's another purpose. That's why I would talk about molecular mechanism today about that, okay? So here I will show you one slide. This paper published 2013 is very good paper summarize brain structure and secure networks. Brain structure you can detect by different tensor imaging. It's a routine, even a lot of work for now. I know one investigator in multi-center, Sushi, she's developed connecting 2.0. It's amazing technology. Look at that. Another function. fMRI, REST fMRI can look at one location, how to commute with another location. This is exactly technology for brain network measurement or network. But okay, if we just look at DTI or function connectivity, it's hard for us to understand what's the exact problem. Okay, how you boot signal, spontaneous activity, or some others task fMRI, what's mean about both? So that means a lot of people work on detail about phase origin. You can use MAG, EEG measure electric signal, or you can use PET, look at some sensor. Just we mentioned about PET compared to MAG. It's amazing story. There's a lot, okay? I do not talk about too much of PET from some others, but really I want to mention directly to next one is whatever you want to do, depending on your person or your master, which one you want to do that. Another, for me, I'm working on MRI, MS for many years, probably more than 20, but really we still think about what kind of marks or how can help diagnosis or what kind of target engagement you can monitor. Then you can monitor disease progression or access treatment. It's very, very hard for us, right? This is another I want to mention, but this also is our target, eventual target. I want to reach that game, but really it's very hard to do that. Okay, today I want to give you some example. How can do this kind of work or can fix this problem is helpful for that. I want to give some simple experiment. First is working memory, right? Yeah, because of cognition. Yeah, psychiatry or some others always has cognition problem, working memory problem. So if you do task it for my, here I showed our experiment, I'm McLean suite Prisma scanner. You just ask a subject do some work memory test, okay? Then you can see some region is inhibited. Actually, that inhibited region is related to default mode level. And some region is red color. You can see that region is excited. We call that is activity region. That is most close to executive network. Okay. So this experiment is very routine. It's easy to do. Yeah, we do a lot of subjects. It's most very reliable. This we call activation and deactivation. Okay, right? So then you can see if we do this kind of work for schizophrenia patient, yeah, people population this paper, you can see whether anti-activation or deactivation is become smaller in schizophrenia patient. It's not excited too much or it's not inhibited too much. Okay, yeah. So this one thing I want to. Another, if we want to understand that, we want to figure out why excitation or inhibition is more small. Let's back to some basic neuroscience about schizophrenia. For schizophrenia, people mostly think about MDR receptor hyperfunction in schizophrenia. That means, we know neural communication with each other. You need neurotransmitter, at least glutamate, right? Or GABA, some others. Another, you need energy support your axon, sending information from one location to another location, right? Yeah. So MDR receptor is glutamate receptor. This play a key role in schizophrenia patient. Yeah, this is well accepted and approved by a lot of molecular experiments or say by work start for now. MDR hyperfunction. That means MDR receptor cannot pick up too much glutamate. Okay, so if you think this is true, immediately people think about how can we do experiment? We just mimic a condition of schizophrenia. This experiment did in year group is very interesting. I like this paper very much. The reason is when you do health control, just infuse glutamine. That means glutamine could bind glutamate receptor too, or maybe because schizophrenia condition, right? They ask here subject do work memory task too. You can see here placebo, that means low glutamate information, and red color ketamine infusion. You still can see both signal, whether positive signal we see activity region, right? Or negative both signal, that's negative we happy region. Both is combined too. This gave us very good evidence. Okay, neurotransmitter glutamate or receptor at least play some work in this schizophrenia model. Of course, you can do patent research, do this kind of work too, but it's very expensive. I would like to say that one for patent, right? So then this we also think about it. No, classical for my right. Another side is resting for my. Yeah, okay. I think this image there a lot of people know about this story. I won't do not talk about too much detail, but I just quickly mention about it. If we just put a seeds on MPFC, right? Then you do whole brain correlation analysis. You found some region like here again, default mode level is positive correlation, right? That's showed here is a warm color, but around region correlation is this value is negative, right? We call this anti-correlation, right? That's in central executive network, CE, right? This is called anti-correlation. This experiment also is much reliable. For me, I really want to use some reliable technology to test some basic problem or basic neuroscience. So here you can see in schizophrenia, also is very clear, this anti-correlation also is combined. That means red color is disappeared, or you can see not too much, right? So, okay, now we turn off, we put together activation for task for my or creation, anti-creation for resting for my, both is compromising schizophrenia patient, right? So what's the problem? Why? What's molecular mechanism underlying to drive this way and describe the brain security, right? Okay, so immediately we think about measure GABA glutamate, okay? So we just choose two regions. One is MPFC, another region is DLPFC, okay? MPFC is harbor in default mode network, and DLPFC is another very important node in exact network. Why we just choose region? Of course, I want to choose more region. The do is sweetie sensitivity issue. This experiment is combined from my task from my and rest my together and with glutamate and GABA measurements, time limitation. I cannot choose the bottom model, right? If I do experiment at 70, so of course you can choose 4. If I'm not sure, a lot of region you can choose, right? Okay, so then I will say, how about first GABA modulate local neural connectivity, right? I show these slides. Then you can say I measure GABA in MPFC. Then I analyze region. That means in the local region, neural communicate and correlation each other. You can say GABA and glutamate ratio has strong correlation with this value in the same location, right? But I would like to say here, major play game is GABA. Glutamate just contribution big. Small, it contributes, but it's small, mostly GABA. I think this is not difficult to understand because GABA is local neural inhibitor. It inhibit local glutamatergic function first, okay? Then this glutamatergic function could project another region like DRPC, right? So then my question, how about GABA you measure in MPFC? Modulate function connectivity between MPFC and DRPC. That means anti-correlation, how to modulate anti-correlation, right? Then, okay, we did this experiment. You can see here, GABA and series ratio in MPFC and strongly have correlation with anti-correlation between MPFC and DRPC. Okay, this is a health control. Okay, this is health people. Okay, yeah. So then this is we call okay again glutamate tergic function in MPFC projected to DRPC, right? Could project to another region. This also we talk about neurotransmitter in one location and remotely modulate another, which is long distance modulation. Okay, right? So, okay, so how about in patient? Okay, I test this model in 2 models. First, the patient is first episode of schizophrenia, psychosis. Another is major depression patient. You can see here, glutamate GABA correlation with anti-correlation also is disability. It's low sonic correlation with a lot of finding that one. Okay. So another, I want to talk about the factor again. How about we follow patient a long time, 1 year or 2 years follow up, right? We talk about at baseline, anti-correlation in schizophrenia is combined. You cannot see too much color here, but also relative GABA compared to health control also is come down, it's decreased. This is not routine error, but people did a meta-analysis or some other, also validated GABA level in the schizophrenia generally come down. But we did experiment is follow up, right? You can see here schizophrenia, it looks anti-correlation come back. Okay, this red color is appears again. But interesting, it looks GABA also is come back too. Okay, right now my subject number is small. We'll continue follow up because always follow up experiment is very tough. Yeah, we wait for more number and see whether follow this pattern. Why this one, since I want to talk about that. Then I will talk about another. Why I spend too much time figure out the GABA inhibition, glutamate citation, what that. But because it's TMS is well established method for brain modulation. Immediately you ask you work on inhibition or rather you work on excitation, right? Yeah, this even for us, we also work for schizophrenia patient too. Our hospital also have resource for depression, sort of for TMS, that one. Another thing for me, I'm very interested because you stimulated brain. I'm thinking about this, some neuron is so silent, it's like sleeping. Okay, now I want you wake up, wake up, okay, come on, okay. Then I'm thinking about another is energy, mitochondrial function energy. Maybe you need more neural energy support, right? Because we always know glutamate, okay, diffuse to extracellular space and then pick up another. We talk about pre-synapse or post-synapse. Always need energy or long axon, right? They can move glucose from one region to another region, right? So this immediately we're interested. How about energy system? Another is neural activity and metabolism coupling. Most is coupling, I would like to say like that way. We spend a lot of time. Actually, I appeared my mentor, Mr. Dr. Wei, I work in his team, spend many years for this kind of work. So how do we measure glucose consumption? How can we measure oxygen consumption? A lot of methods you can choose, okay? Here, I want to think about first proton spectroscopy, carbon-13, or deuterium, or O17, or P31, okay? Here, I just quickly mention about one experiment I did before, is glucose fusion experiment. This experiment we done in animal, but we also did with human study too. Yeah, okay, this one I would like to mention about is natural glucose, okay? It's not just certain infused labeled glucose, it's natural abundant glucose, okay? You can see here, I'll show you first is baseline brain and glucose level in the rat brain. After you just give a bolus, okay, infuse glucose level, increase glucose levels in blood. But of course, you can continue measure blood level glucose. You can also continue measure brain glucose level. Okay, I show you slide 40 for animal brain study. A is baseline, B is after infusion, C is difference. You can see glucose level. D is pure glucose. Okay, you can see here, right? But of course, I would like to say this experiment is quite tough because glucose is very small signal. Another concentration around 1 millimole. Okay, but anyway, after that, you continue measure or monitor glucose level in the brain or in the blood. You have model how glucose wash out and in, then you can figure out glucose consumption rate. Even you can measure some other parameter is the glucose transportation constant for this experiment. Okay, this one talk about, of course, another you can do see certain labeled glucose, okay, for this one. I would like to say this is tough, the experiment I did before. It's very, very tough, yeah, C30. This one, we want to figure out how about your neural activity or neural energy support your with different stimuli response. You know, here we do experiment simultaneous 4 hertz and 8 hertz, just change for different size with different frequency, but it's simultaneously. Another, we want to watch this 2 location, okay? Left and right side, same experiment. Okay, here, I do not want to talk about too much detail about this experiment, how to do this one, but I want to show you, say, certain labeled glucose, then you can pass T-shirt cycle, then you still can figure out how much glucose labeled glucose could convert to, say, GRU, say, certain labeled GRU, okay? So generally, you can say here, also is both blood flow and energy. T-shirt cycle still is quite coupled together, okay? Yes, this one. Or of course, I would like to say this experiment is very tough and it's very expensive, right? For that one, I also want to mention about the O17. You can measure oxygen consumption ratio too, but that's why it's expensive compared to 13. Frankly, it's very, very expensive. Yeah. I would like to go some cheaper. Yeah, okay, cheaper. This phosphorus C13 is natural. Okay, you do not do anything, just lie down scanner. We do not infuse anything. Then you do experiment, you can measure phosphorus signal, okay? P31. This is experiment I showed you at the 70s, a long time ago, okay? So here you can measure substance level like a phosphorus creatine, its energy reserve or energy buffer. And ATP is direct energy resource can be used by cells. You can also measure inorganic phosphate. But I would like to say phosphorus creatine and ATP and inorganic phosphate is chemical coupling system that is catalyzed by criticalus and ATPases, okay? Criticalus that's related to energy transportation system and buffer, and ATPase is related to energy generation and utilization. So again, I do not want to talk about too much detail how to do that work. I would like to say all this for chemical exchange reaction rate for reverse is measurable. We did experiment on this one also. And most conditions, this also is a balanced situation, it's equilibrium system, okay, for that one, all right? So another, I want to say, magnetic transfer phosphorus 16, right? You can measure criticalus activity for that one. But usually people talk about it, experiment is too long, it's very hard to do, okay? But really, I would like to say most you can combine with CSI, chemical shifting. I will show you animal study and line 4. This is for hard study. We did one-dimensional CSI. You can say even you can say up, down, then to hard muscle, then to chamber signal one. Also, you can do saturation. First one is 1D control, low saturation. Third column is for saturation. experiment for that one, right? So even you can do 3D CSI for human brain. This also paper published in 2012. We do that. Okay, so here you can say 3D says we can do a lot of experiment. Okay, but anyway, this is quite a complicated story and quite complicated formula and figures. I do not want to spend too much time. But okay, generally speaking, I will just give you overall picture, magnetic transfer, how it works. Okay, that means when you set gamma ADP, okay, so this runs, you can see magnetization phosphor creatines come down. This because chemical change rate between phosphor PCR and ADP is catalytic precast. Another, you also can see phosphor PCR1 signal is still come to that's catalytic about ADPases. Okay, then if you quickly do a calculation, how much energy generated from oxyphosphorylation? Okay, that means ADPases activity. You can see one day our brain consume about 10 kilograms. Go ahead. Yeah.
Like when you, with this magnetization phosphor, so you said you saturate gamma ATP and then the amplitude of the phosphocreatine peak decreases due to the enzymatic reaction. That's what you probe in this experiment.
Yeah, we do gamma ATP saturation, right?
And then the phosphocreatine peak decreases because of the enzymatic reactions that transfer phosphate group from. That's what you probe.
That's because chemical reaction between PCR and ATP.
Yeah.
Yeah.
My question is like, I mean, they are pretty close in like chemical shape. Like, I mean, it's you have to be really very RF selective, like then very narrow bandwidth.
Oh, I got you. That means you call off resonance effect, right?
Without affecting directly.
You're right, exactly.
So and I think it's impossible to avoid. So how do you correct for that?
It's very good question. We have very narrow saturation band. I will show you.
Saturation band, you still there is a tail. You still so you have to correct for it. Do you correct for it? Or
I could talk to later. But really, first thing, we have very narrow saturation band, only around 150 hertz.
Yeah, but
so right now
you have also zero chemical shift or a lot of things that, and there isn't. I mean, there is nothing like a rectangular box. Like there is always a tail that would hit. Like I think you never get like a perfect profile.
I will show you. Yeah, actually, that's we call another. We develop very good method. We call bistro. Good thing is first thing, bistro power is less, not too much power you need. Another thing is we have very good shape if RF pulse. This shape RF is narrow and very sharp. Yeah. So, but anyway, I have something correction for this one.
I think, do you also, I mean, you can correct for it, even if you hit it a little bit, the phosphocreatine, probably the effect is less than what you want to measure by enzymatic reaction. You only have to correct for it. Do you have that? Do you do that?
We have that. Yeah, okay, good. Okay, this is for ATPases, you get 10 kilograms, it's very large number, right? Another way, if you look at catalysis activity, that means energy buffer. If you generate a lot of ATP, you have to convert to PCR, save in bank, energy bank, right? If you have enough, you come back. This is close to 40 kilograms per day, almost half 50% of our body weight is huge, right? Yeah. So this one thing I want to show you. Another, I really want to know, we did it from how about neural activity at a different level, how much energy you need, right? So then we did animal study, we just do anesthesia experiment. It's light anesthesia, you can EEG spike and isoferrin is very clear. Then we use low dose orbital, mid-level anesthesia, spike, not too much, less a little bit, right? Then if we completely shut down EEG spike, okay, we call isoelectric condition. Most new low neurofiber situation like that, right? Then you can still measure how much glucose burn. That's glucose consumption rate, right? Another is CK activity at ATPases. So you can see, as these three condition energy, each other has very good correlation, and correlation with EEG entropy. That's the index of neural activity, right? Yeah, this is actually I will show you at higher anesthesia condition, highest EEG completely shut down, your energy mostly reduced around 50%. That means at least our energy, 50% used neural spiking. It's quite high. Actually, our brain like a 21 bubble every day. Whatever you want to do, at least 21 bubble is sharing. It's working on your brain. This is huge energy support, right? Then we back to think about how about disease condition, right? That's why we do schizophrenia, still measure magnetic transfer, measure energy system, right? You can see here, creating callous activity in schizophrenia is down, it's decreased, it's significant with substance and secondary reduction. This experiment I want to mention about is 40. Okay, 40 slide, which is later for the one, right? This means schizophrenia patient may kind of have some problem. Okay, so if this have problem, what will happen? We talk about it, probably have some condition, potential high energy demand, right? For example, you work hard, then you have some problem. We test them, we do function FMS, just to give you task or some other stress condition, okay? So here, I would like to show you two papers here highlighted here. One is the function MS in schizophrenia, another is depression story. Even we measure GABA glutamate for the model, then we can see some different story like that way, okay? So here show you. So another thing I want to talk about it is how, before we already talk about it, task for my rested for my energy neurotransmitter and by the counter function and energy system so far. But so far, we have to answer another question, how about neural communication with different location, another region, right? So here, I want to mention about, again, our energy measurement is the MPFC. We want to say how about MPFC energy modulated long distance neural communication each other. That's why we did correlation analysis. You can see here, CK flex and the function connectivity, whole brain correlation. Then you can find still is very interesting in default mode network, like PCC, post-arterial synchrony, still has very strong correlation with energy. CK flex, okay? In his control, you can see this is here I show you green color. It's very, very strong, right? But in schizophrenia with bipolar disorder, this is compromised, do not have very strong. Interesting, you can see bipolar disorder looks a little bit stronger compared to schizophrenia. But good thing is bipolar disorder also energy compromise also smaller. Schizophrenia energy problem compromise much higher. Okay, this was since this we talk about is intra-network, default network, because MPFC, PCC are origin in the same default network. How about inter-network? Okay, that means we talk about central network and creation, right? You still can say here energy MPFC, right? Have very strong creation with relative sense value. That's anti-creation number, right? But still compromising bipolar disorder and schizophrenia patient. Okay, so here I would like to say first take-home message popped out as the first take-home message. Energy demand associated with neurotransmission would be critical for large scale and long distance neural communication. They should be very important for better executive and cognition functions. Okay, this probably is our target for therapy. Okay, yeah, this first message for home. Okay, so then I would here give you a model. We just talk about redox system. Sorry, we just talk about made counter function and glutamate hyperfunction, right? MDR receptor, glutamate, we talk about that. Another people think about in schizophrenia, redox degradation also is very important for this one. Of course, redox degradation and mitochondrial function sometimes is tied to each other, okay? So now we talk about whether we have method to measure redox ratio, okay, AD, ADH. This picture I show you here is very fundamental neuroscience or biochemistry science, right? How energy generate, how energy consuming ADH system, okay? But here I would like to show you for phosphorus phosphate, right? A long time ago, people pay attention about ADH, a signal is here, but this signal is very small and lowest level is very hard to separate if SR is not enough. Another is how can we separate AD and DH, okay? So here I'll show you spectrum required at 40 mechanistic scanner. P31 without protein decoupling and with decoupling. You can see with decoupling, spatial resolution is much, much better. But of course, you can see this spectral SR also is wonderful. Okay, that means you should have very good SR, very good coil, and very good shimmy. Then you can reach this goal. Yeah, okay. Now then how about synthesis? That's why we come here. I want to build up this experiment together too. We do 70, so of course sensitivity should be much better compared to 40, right? But I show you here it's doable. NADH is measurable, right? But which one is better, 4 decoupling or 7? Well, that's we could test in the future. But yeah, you make a decision, right? Like that way, okay? So another, how about NADH in sclerosis and aging? Okay, here I will show you. You can see immediately this slide. You can see, when you become older and older, redox ratio is decline. Patient is sclerosis decrease more compared to health control plantation. I would like to say this redox ratio decline is natural plantation. It's naturally. You do not have any choice. We older this one, and this redox ratio is come down for our family or some other people daily improvement. Most is driven by NADH goes up a lot. NAD plus also decrease a little bit, but slight decrease. Most is NADH goes up a lot. Okay, this is what mention about that. Another, I will show you here this curve. We just do a linear fit quickly for chronic subject or older subject. But how about early stage, younger subject? Right now, I do not have too much data. We'll continue to collect this data to this experiment. We should probably is U-turn. It's hard to say right now. I'm not sure what kind of pattern it is. Okay, so then I would like to say, okay, we talk about redox part. It's easier to understand why redox has some problem in the some wrong with mitochondria, right? Because when mitochondria generate ATP, you must have NAD plus, give electron, then electron power electron mitochondrial membrane to generate ATP, right? If this system with redox has some problem, you should have some problem maybe with antioxidant, with oxygen stress. So how about GSH level? Another, we just talked about before, mitochondrial function, bioenergy, so we can do magnetic transfer measure, right? So now I would say we can do it. So we can put everything together. That's why we designed a surface coil, 2 channel, proton and phosphorus in the same experiment sensation. You can measure glutathione, GRU, glutamate, we just talked about it. To minimize the T2 effect, we use TE, short time 10 milliseconds, develop a spatial sequence. Another phosphorus signal also is quite here. I just show you NADH, but of course you can combine magnetic transfer together. That's fine too. That's why we, like say, we put everything together in the same experiment sensation. You can compare to each other, protein channel, phosphorus channel too. Okay, for that one, right? So now another piece I want to mention about it is neuroinflammation. Just one moment ago, I show you slides. Probably I come back to one. Okay. So here we talk about immune stress pathway. In schizophrenia, neuroinflammation also is very, very important for this one. Okay. So that's why here I will show you. So do we have some evidence to show inflammation in schizophrenia? We say we have. So here I will show you first result of study. We do measure MIS, my here. You can see comparison control. My astral signal goes up. Also, GRU also goes up too, yeah, compared to his control. Okay, so here I want to quickly summary together. So far, I think this is a second message to whom schizophrenia may be characterized by excessive neurotransmission associated with elevated energy consumption at early stage. Because you work very hard, overexciting excitation, this will gradually downregulate and lead to toxic, right? Decreased p-value or some other oxidative stress, right? Do we have some data show this concept? I think we have. I'll show you here. We put together about the first photo and Crohn subject. We put everything glutamate receptor GRU an energy system, including chaos, even substance level and pH value. We also put redox together. You can see here, right? It's energy pattern, how to with this progression. Another thing is how about proton? That means high risk before onset scapula, right? Actually, we'll work on data collection for this one. Okay, so right now, right? So another I would like to mention about immune stress, right? It's not only for scapula, but actually in AD, Alzheimer or aging or so is quite well known. I just did meta-analysis, look at how about measurements right now in this field about the spectroscopy, link energy system or link to neural information to whole system, right? But still you can find my estimate goes up too, right? Another thing I want to show you, just one paper published in this March this year in Nature, they indicated the scapula and aging may share a common biological basis. Okay, go ahead. Yeah.
Here.
Yeah.
But you know, like schizophrenia, usually the symptoms of schizophrenia decrease with age. Like this is a problem, like a disease that's of young age, and like people that are usually as early as 15, but in the between 20 and the 30 is the peak. And that's where you see the most cases of schizophrenia. And then the gravity of the symptoms and everything, they decrease. And then when they are 50, 60, they have less acute symptoms. And so how does that square with this hypothesis that aging and schizophrenia work? I need to discuss this with those.
Great question. Great question. I will talk about later. Yeah, in my slide. Yeah. Okay. Okay.
No, so like it could be that, for example, the patients with schizophrenia, let's say they may have an older brain already when they are 20, but then they should get progressively worse, right? It's like let's say when they are 20, they are older than somebody, a healthy person that is 20, but as they age, they shouldn't become better. They should become also worse. Right?
You're right. That's a very, very important thing. We talk about heterogeneity issue. Heterogeneity. Yeah. Okay.
There are the people that don't.
Yeah. 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 My slides probably talk about later. Okay.
Yeah.
Yeah. Okay. Sorry. That's why this slides you can say, right? So that's why we really wanted to do, because we talk about aging with scapula shares some basic biological basis, right? How about aging effect with scapula? We talk about that. Okay, so actually people really found that scapula died faster, died early compared to healthy people. You know, in America, generally healthy people probably close to 80 or so, scapula close to 65 or even early. Why? What's the problem? Another, you talk about maybe scapula recovery, oscillation, conditions poor, then come back, recovery, then worse and worse again, right? So really for us, it's very hard question. That's why we have another grant just coming in, want to do this experiment, longitudinal monitor. This project, we monitor patient from 40 to 70. Yeah, 10 years, 40, 50, 60, 70. But each 10 years window, we follow 2 years. Baseline 1 year, 2 year follow-up, right? Then that's why we want to work with you, do this experiment together, see what kind of pattern we have. Okay. Yeah. So here I want to show you about that one too. So another, everybody want, how can we slow down aging effect or some other? How can we do some intervention? Currently, a lot of methods is available. So here, right? I just mentioned about it. But really, recently, I think read our survey with ND plus booster immediately, because you say ND plus is come down, right? Another ketogenic food is quite interesting too. I want to mention about that. Of course, some other browser. But really, for my understanding, usually this kind of work is not very successful. Yeah, you can look at nature. Why? What's the problem? I would like to say, probably we need a marker. Why is working? Why it don't work? You have to answer, right? Why individual, this person is better, another person is better. So really, we have going to deal with this one. So one thing is that we do AND plus supplemental is precursor, right? So our target is redox ratio AND over ANDH. That's our target. If you say you give this submental is working, you at least should say you probably could elevate readout ratio, right? Good job. So even you can elevate readout ratio, but you cannot guarantee it's really work because readout ratio have to make mid-counter function better. Well, that's why we measure mid-counter downside effect, right? Of course, so you can measure GSH. If mid-counter works better, if it's higher, antioxidant level probably would be better, right? That means we put together marker. That's our goal. And long term, if this really work well, then we can check whether this could improve cognition function or not, right? So this is really a lot of work on project is one understand biomarker, it's guide your treatment or it's work or it's mechanical. What's the problem with that? Whether it's work, right? So again, we back to this guy again. Yeah, the biomarker is very important for that one. But we have to think about it still is very hard. This is patient heterogeneous, very, very difficult to do. Here I show you one project is we do function connected analysis. Again, I want to show you 1 year follow-up. If we put this 87 subject together for functional analysis, less than 1 year follow-up compared to baseline. But if we use some method, machine learning, automatically separate different groups, B and C, then you can see some difference. Okay, this is preliminary data. I'm working on continue for this one. But really, I want to emphasize heterogeneity is very hard for neuropsychosis disorder biomarker or some other treatment response to look at. Okay. So here, I just quick mention about neurosecurity network structure. Sorry, energy transmitter and connectivity. But really, I do the structure. Fiber also is very important. E-signal sent from other have to allow axon or myelination, right? So really, DTA differential is easy to do. I just show one picture. I do 2001. It's ugly. It's frankly, it's quite ugly right now. It's 5 minutes. In 6-4 dimension, you can do very good orientation fiber tracker for that one, right? But I will give you another example why brain is very complicated because compensatory pathway or plastic issue. Here, this project is collaboration with China, who has some subject low cortical level. Some subject has only partial cortical. You can see here, right? But basically, all these subjects, they told us basic function and the direct function looks normal, not a big problem. Okay, so that's why I really want to understand why structure, what's really clear, what's can do for that one. So then we did detail, structure connectivity analysis, and also we do function active analysis. So generally speaking, overall, the brain structure and connectivity or function connectivity has not too much big difference. But really, we found some small region really enhance stronger whether connectivity or whether structure connectivity. Okay, so this is another we talk about very important concept is conceptual pathway to plasticity, right? Another I really want to do a structure problem. How about the detail mining, axon issue or some other issue, right? But really, we would like to say we always everybody low, MRI, right? A lot of parameters you can do. We call parametrical imaging, right? So for us, we're really interested in water, MTR, T1, T2, right? ADC diffusion. Even we're interested in AA diffusion because AA only tested in intracellular space, not extracellular space you can find AA, right? How about we put this together? This is very preliminary experiment we want to figure out. So here I just show you quickly. First, I want to know whether really water T1, T2, or ADC has difference in schizophrenia or patient or AA. But so far you can see really you have some difference. Okay, one example T2 in schizophrenia, water is higher. Okay, water T2 is higher, but AA T2 lower. Okay. But MTR, you can say all schizophrenia or bipolar disorder is lower compared to health control. Okay, but in diffusion, only schizophrenia compared to his control goes up a little. Probably this reflects some pathology or pathology condition like myelin demyelination at early stage, then gradual damage to axon. But really, this also is hypothesis. Do not have too much data prove this concept. But really, it's very interesting for us in the future. I will talk about that. Okay. So again, MRI, MS, marker treatment, whatever we want to do, I would like to say this is my last message to him. Fuse multimodal neuroimaging, clinical data. Of course, we have some clinical data, biography information together. Then probably in the future, we want to fuse everything together with machine learning. This is quite hard. Everybody wanted to push this direction. For us, we want to do this kind of work too. Okay, this is the last slide about my talk. But finally, I want to say this is teamwork. A lot of help from my colleague, McLean. Tosanger is my most collaborator for schizophrenia, bipolar disorder. Some other people in McLean too. At University of Minnesota, my mentor Wei Qin, Xiehong, I work here. A lot of people work for us. And Tufts Center. And my MGH, of course, we just talk about Ovidio and Eva. Some other people, Jason is here too. Even a long time ago, he passed for Koyo ever since. So here, okay. Okay, I would appreciate funding from H, Lhasa, and some other resource for this one. Again, I greatly appreciate you, everybody, and I really like this opportunity to work with everybody together. And we actually have one grant to work on STEM slide here in Martinos Center. Okay, that's my everything. Okay, thanks so much.